and I will turn it over to you, Bala.
Mm. Okay, thank you for the intro, Hannah. It's my pleasure to meet some of the known people here virtually, and I love to give a talk on what I experienced from the academia to the transition. So, when I was a few years ago, when I was in academia, the publication was the big thing for me. Whenever I saw my name on the paper, that was more exciting, and then that meant like a success for me. But when I moved to the industry, the things are a little different. It's all about timeline, deliverables, and then the meeting the making impact on the product at the end, for if it is being a cell therapeutic product or gene therapeutic product or food industry. At the end, the product is the product was the success. So the things when we are transitioning there, the things are different. That's what I thought. The I can give a talk on this one from publication to pipeline. That drive me to choose this topic to talk. Nice to meet you all. And bef- when I go, one second. Okay. So a little bit, I will give you background about me and the roles I did in the industrial jobs. And now I'm in the transition stage. So I'm looking for the more better job. But that's why I said the past few industrial experience I will share with you the roles here. And the purpose is to show how the transition happened and everything I will introduce to you. Like my journey, I originally from India. I'm an international student. I came here then I went to South Korea for a PhD. I got an excellent lab. I got good training, good publication there. Then I came here as a postdoc. I stayed here as a postdoc or glorified postdoc for 10 years on visa. Then I waited for my green card. I applied. We got in the topmost category. And then once I applied got the green card, then that was the time I wanted to go for the transition. Uh, so it took me like 10 years for, after my PhD. But everything gave me all the PhD or postdoc labs gave me a lot of knowledge and so many area of expertise I got that. So that helped me to ended up in the even my first job I was getting as a senior scientist. So everything helped at the end. So my main focus was in the research side was mostly molecular endocrinology and metabolism, and mainly cell and molecular biological approaches I have done that helped me a lot to fit into any kind most kind of roles in the industrial jobs. And then when I was initially I was in academia first, and then I moved to NIH for like 4 years. Then I came to the UC Denver here, our Anschutz campus, stayed there for 5 years. Then I moved to the gene therapeutic field. That was a like contract job, and then after that I moved to the cell therapeutic field. So that was very good job. I really love that, and somehow they then after few years then again I have to move to the food industry, pet food industry. So the transition happened, and then we may end up in contract jobs and full-time job. And that job, even though there is short term like few years or 1 year, so still it gives we can build our CV, and it also help us to find another and the next job, more better job. So everything gave me some experience, and everything helped me a lot. And then when we look at the this life science ecosystem, so academia, most of the people they gets idea from the academic researchers like publication that give us a direction to choose which way we can go. Like even in biotech company or in pharma, people choose, okay, this direction is good, this many publications are driving towards this direction, so we can focus on this part and we can try to develop some product based on that, like cell therapeutic or gene therapeutics or any pharmaceutical product. So academic research is very important, I feel always. So and also uh, everything is interconnected. Like biotech field, they give as instrument diagnostic tools, and also the even the reagents for our own academic research, everything is there. And the if we are going to the pharma, there are few kind of pharmaceutical departments are there, like cell therapy product, gene therapy product, or drug product directly. So each of them depend on the publication, what we do in the early research during our academic research. That so everything we have to complement each other. All the and then all of this food industry, everything, all this industry are like kind of supervised by the FDA people. So everything is good. Is it good for safety and efficacy is good? If it is going to the people, it will improve their life or benefit for their disease or something like that. So they keep eye on it. So regulatory department also very important, and everything is interconnected. If we are losing uh, grants or research funding in academia, eventually or later it will impact the other industries also, because everything depend on the basic research. So and some of the manufacturing partners are there, like R&D and manufacturing partners, like anything start with C, like contract research organization, contract development and manufacturing organization, contract testing organization. They are also a good when I first moved from industry academy to industry, I went I went to the CDMO. So that's also a good stepping stone to start our industrial experience. That's a contract job. It's anything with C is very fast paced, and then everything we have to follow hard copied SOPs. And if we are missing like few, if they're telling, like for example 20 minutes plus or minus 2 minutes, means we have to be within the 20 to 18 to 22 minutes. Otherwise we are there is a deviation. Then if there is a deviation, it's a big thing. So they will go through the investigation, a lot of things. So we have to strictly follow the written SOP in case of the QC setting. Like if you are working in the QC jobs also good. Like if you are going as associate scientist or scientist, and then QC jobs are very good. We'll be doing only one kind of work, like qPCR, for example, qPCR or digital droplet PCR or ELISA. We'll be doing that one only, but everything is hard, written, hard written. We have to follow everything. At the same time, contemporaneously, we have to record what we are doing, when we are doing, what is the time, like 7:15, and then 7:30, like that. Everything we have to record. That's how FDA audit them. If anything goes away from the hard written protocol or SOP, they'll think like a deviation, then it's not good. So even only one experiment um, we have to focus on that experiment, follow the SOP, and then after that, lot of people review, internal review, and then QA people will review also, like quality assurance people also will review. And then at the end, we can complete the protocol. Oh, everything is done. Then this method is validated. It is good for going to the clinical trial or something. That's what so the contract, uh, like CRO, CTO is also very good start for the first transition from academy to industry. Like when I moved to the industry, the aspects, if we are comparing here from academia to industry, like the core motivation in academia, we feel like always curiosity driven discovery. So we are always curious to test a lot of things. But in industry, we cannot, even though we think like we can test this one, this will make scientifically more good to test, but we have to go through the tier approval, everything, and then mainly it is deliverable focused. So they focus on the product at the end, not the curiosity. The curiosity driven. It happens, but has to go through the multiple um, steps, and then like management, then their management approval needed for that. And then the culture is in in academia, we can openly critique, and then we can debate on whatever the data, or the data looks good and not good, it can be better or something like that. But in industry setting, when I moved there, we cannot give a direct critical open critiques there. So I have to follow the team and then hierarchical structure, so this kind of thing. And then recognition, and in academia, publication or grants are obviously, and degrees also for anybody who is waiting for the degree. And but whereas in the industry, it's all about the team based teamwork. So mainly people focus on we focus on the publication here, but in the industry, product at the end. And this is about one of my role in gene therapeutic the field. So where I was working in a QC, like I said, QC is very strict. We have to, and then very strict, and also all the SOPs and then procedures are made to minimize the risk. So we cannot take risk, because already people in the development, analytical development or product development, they make the protocol or SOP, and then it can be transferred to the QC. Then QC, the one who tests everything. Okay, this gene therapy product like vaccine, for example, vaccine, their titer is good. This is within this acceptance criteria. All the acceptance criteria are met, then they are good to go for large scale manufacturing, or it can go to the clinical trial. So we have to strictly follow GXP. They say like good manufacturing practice, or good lab practice, or good document practice. So everything should be documented. We cannot simply do our research and then we can do few days after we can write down about that one. No, everything should be contemporaneous. So this gene therapeutic experience is different, and if we are working in QC setting is completely different. And then next I move to the cell therapeutic company. They focus on the cell therapeutic products to combat the solid and liquid tumors like lymphoma or solid tumors. So that product has to be validated, because the product will have, uh, even though it is a cell therapeutic product, there are multiple gene editings are there, and then we need to confirm whether it is edited properly. Our gene product is the right product by testing the identity okay, this product is the right product, and then it has no other impurities from the host cell or anything. So this kind of testing I was focusing on, basically analytical development. That's like one step before the QC. And then once we develop this and from analytical development, we can transfer to QC. Then QC will double check with the double check our protocol with more strict acceptance criteria, and then when they meet that acceptance criteria, then it will go to the clinical preclinic from preclinical trial to clinical trial. So that I learned a lot from that company. Also, mainly I was focusing on ddPCR based assays. Even though in academia I didn't have any ddPCR experiment, I had qPCR, like more than 10 years of qPCR experience, and then I I can easily adapt to this ddPCR. And then the training's also different from in academia. We can follow multiple papers or materials and methods, and then we can set up a protocol. But there they have a hard written protocol, and then someone will train first. Even though I worked in ddPCR for more than 2 years, if I my next job, if I have to do my assays based on ddPCR, then someone will train me, and then I have to watch them when they do, like it is called like observe run. And then uh, I have to uh, do the run, and then the trainer will watch me. That's called supervised run. And another one is like I have to do everything independently, and then at the end, trainer will check all the data, everything is met, all the acceptance criteria are met. That third run is called like independent run. So only one assay we have to do like three different run at least, and then only I'm qualified scientist for running that assay. So there uh, every department has a little bit different. Like if I go to this, when I worked in the food industry, it's an early research experience. It's a more earlier development, but for that we don't need to have a strict SOP, everything. We don't need to do anything. uh, just we can do almost like a academic research setting. Like we can pick up a topic, we can work on it like organoid development, and then we test that. After developing the organoids, I use that organoid to test the efficacy of the ingredients, the past ingredients in the food industry, are the novel new ingredients, so we can test their efficacy, this kind of thing. So this one has little bit more freedom compared to working as an analytical development scientist or a QC scientist. So that's what I had, like three different roles: one in gene therapy and cell therapy, and then third one is in food industry. So everything gave me different experience, and I got exposure to different kind of cutting edge technologies. Like here I did like nanostring, and also lot of sequencers. We have like PacBio sequencer and then NextSeq 2000 sequencer. So even though I didn't have, I used to, in academia, I used to submit my sample for sequencing to the core lab, but there I was running, I had to prepare the sample, and then at the end I have to run the sample also in the instrument. So from the beginning to getting the data, uh, so I have to do everything by myself. So that gave me more experience and knowledge there. And little bit, if there in the industry there are some unspoken rules, like if the smartest person in the room isn't the one who always wins. So just whoever adjust quickly and understand the people and try to be in part of people, they are the most uh, most successful people, I would say that. And for unwritten norms and like what's on the paper or procedure, but a lot of unspoken practices are there. Like we have, even though uh, we we can openly discuss everything people say, but we cannot openly ask like one step higher. Like if my manager's manager, I cannot reach, or anyone cannot reach directly, avoiding the immediate manager. That kind of things are there. And then we have to like when we are even though we think some of the presentation or ideas are not scientifically relevant, we cannot give a direct critic on that also. So we have to diplomatically speak and talk and try to be with the team, and then not to um, uh, like any normal critic also people take it personally. So this kind of some unspoken rules are there. So uh, even though we end up getting the chances and staying there and adapting to the industrial setting a little bit challenging. That's why I'm giving some examples here. Like in our in academia, creativity, we always look for the creativity which drives the discovery. But in industry, conformity. So if uh, 20 people are there, if 19 people are telling one is right, so the obviously the 20th people also will say even though he has difference in opinion and it is scientifically different, we have to go along with the team because they want to focus on the deliverable on time. And so that kind of things are there. And then independently we can critics also. uh, we cannot we cannot make the critics very direct, so we can do something diplomatically. But what about we have to wait for the time or when to do or how, whom to raise the critics. And that's the key thing I'm also learning. It's not it is not easy to be it's a little bit challenging because in academia we love open critiques and then we love open discussion everything, but there we have to watch whatever we are going to criticize or comment or any questions, everything. I have to be careful, diplomatic with that. So this kind of challenges will be there. That's that's normal everywhere. It's not like academia, but we have to be a little diplomatic there. Even though a lot of few challenges are there, but we get good competitive pay, and then we get exposure to the edge technologies. And then if we need that technology, definitely they will bring that technology. And then we for the business people, definitely if we add the business value for any technology, then management and leadership will be ready to bring that technology. So definitely no doubt about that. And then and also I always feel happy that I'm doing my work is having some impact on the product which is going to the people, which is going to help the patients, or which is going to improve the health of anything like animal or any product. So I feel like real world impact is there. So definitely we feel happy, even though academic research we have that impact more much earlier stage, but this one we will see that impact very soon, or soon in near future we can see that impact. That gives very much happiness working in industry. And then obviously career, even though if we are losing job or if we are in the transition stage, no problem, because whatever we worked, like if I worked for 5 years, that experience also matters. That will end up uh, getting a more better job in future. It's not like if I in academia, if we are doing our research, and if we lose that funding, and if we are moving to other one, we completely lose that project. We will start the project new from new lab or new area of research. But here, every in industry, whatever experience we gained will help us to find better and better job and better competitive pay. And that definitely no doubt about that. Oh, that's all. I have very brief one, and then I love more discussion than just presenting about myself and then everything, and whatever questions you have, please ask me. And then also, if you have anything uh, to know more about me, also I'm happy to share with you. Thank you, all of you.
Yeah, thank you so much, Bala. So for questions, oh, I like the little claps. Oh, fun. So if anyone has questions, you are welcome to either type them in the chat and I will read them out loud, or you are welcome to just unmute yourself and ask your own questions. So as people are thinking, I think I will just get us started. Um, do you have just any sort of general advice for people who are in academia right now and who are wanting to make that transition into their first industry job of things that they could do during their PhD or during their master's or during the postdoc to make themselves like more marketable, like maybe techniques or, should they really focus on publication? Sort of what can you do to help you
Mm.
make that first transition?
Now, publication for early research, if you are going to early research like R&D or R&I, people say in the industry, R&I, research or innovation, something, that kind of department, they focus on the publication, how good publication we have, everything. But if you are going to the different departments like QC or analytical development, or because uh, if we look at a cell therapeutic product, they may have like 5 to 10 product in the pipeline, but few hundred people will be working. So each will be a lot of scientists will be in R&D, and then some of them will be in analytical development, product development, like that. It depends on the department where we are going. If we are going to the early research, then publication matters. If you are going to the other roles like QC or analytical development or product development, then exact uh, skill set matters, because if they want ELISA, DDPCR or qPCR, we have to have that experience, and we don't need to show whatever we have all of them in our past experience. Just we can modify our resume only for these three, like qPCR, DDPCR, for example, I'm telling, and ELISA. And that way people think like, okay, this is he or she is the right candidate. So we can do that. If people are doing masters and if they are looking for this transition, the good way is like they can try for internships. Internships are like 2 months or 1 month. A lot of companies they will offer internship. So if they are going internship, then they if they are building a network there being a part is more the key thing in a team. So if they are breaking the ice, if they are being a part of the team within the 2 months period, then most likely they will end up going to that company, or the company most likely wants to keep them for the future jobs. So they will bring you, they will take you there for sure.
Yeah, great. Thanks. That's great. Great
Yeah.
advice, especially the internships
Yeah.
piece. Love that. Okay, so we have a few questions in the chat. So I'm going to read our first one. This is from Cassie. She's wondering, academia can be frustrating in terms of troubleshooting and getting the experiments and data to work right. Do you find that this is still true in industry? Or do the resources, such as teams and money, in industry provide for more robust and successful research?
Uh, I would say the early research is good in academia. We have the freedom of testing a lot of things, and even though cutting in technology, people say like industry, they boast themselves. We have all this cutting edge technology. I say our core labs are they also have the equal cutting edge technologies. They have the latest sequencer, latest instrument or microscope, everything. So we can access that. For early, uh, for research, it is good academia, but if we are moving to industry, everything we have to follow the SOPs or the management decisions. That's what we follow. We cannot if we have extremely interesting one, and if our immediate manager happen to like that idea, then if he is courageous to move to the upper management, his upper management, then we have chance to do better research there. But otherwise, everything they know, okay, this one, this year or this quarter, we can do this, and we have to focus on this deliverables. So we have to, in industry, all about deliverables and product. But I say the freedom of research is in academia is better. We can use the resources from core labs or other collaborative labs for sure.
Mhm. Makes sense. Great. Great. Okay, we have another question in the chat. This is from Ashish. He's asking, as a J1 scholar, I would like to understand how to manage visa requirements when transitioning from academia to an industry position. I know you mentioned that it sounded like you stayed in academia
Yeah.
until you could get your green card, but maybe you could also speak to, since you've been in industry a while, if you know anyone who's made that transition while still on a visa, and just sort of like the pros versus cons of your path versus a different path.
I was also exactly on same J1 visa for the maximum we can be there is 5 years, and H1B also 5 years. So I use that opportunity to get, but it's my personal experience, personal advice. I use that opportunity to get more exposure to different area of research, and then increase the number of publication. And then when I get good amount of publication, then only it's easy for us, it will be little better for us to get apply for permanent residency, something like that. I would say J1 is not that uh, J1. If we are in J1 visa, industry cannot easily take the people, hire the people easily. If even if you are in H1B visa, academic H1B and industrial H1B is different. So they once we are like if we have 2 years of H1B visa, within that time we can work in industry. But once we are you losing that, then industry cannot sponsor. That's why they have a screening question always when why in whatever application we do. Do you need sponsorship for is are you a permanent resident or citizen? So that way they screen that, and most of the industrial job they they step back hiring people from J1 visa mainly, and H1B little bit better chance they have. But if you have the permanent residency citizens, no problem. We can move anytime. We can try any kind of job like internship, initially or contract job or hourly jobs also. It's fine because some people they look for like 3 months they will pay hourly this much only. That's also important. We can grab that if we are looking for the first transition. We can grab that, or if we are in a big long gap, also we can grab that. Even I would also, if I had to wait for few more months, like some of my friends were waiting, so that time I may think of getting the hourly jobs also. That also add some experience to us, and then that will also help us getting the full-time job in the same place. So so visa little bit difficult with visa. That's what the screening question, first question start there only. Do you need sponsorship? That's what they start. That's why I waited for a long time patiently, but I enjoy my uh, research in academia. I build my CV better and better over the time. Then I used to be a lab rat. I don't lab rat. I don't talk instead other than the science or something. So then I started networking like PDA. I got involved in PDA. I was also managing the seminar series, everything. So that gave me icebreaker, a lot of thing. I came out of my comfort zone, and then everything is good. Every group I learned something good. So uh, so I would say when we are in visa, we can build our CV and networking better, and then once we get that stage where um, the industry they don't need to sponsor, or they don't want to spend some of their time, like HR people should take care of that. They don't want to focus on getting uh, visa for someone unless if it is extremely important. Otherwise, very rare cases we I noticed there. And that's what it's my personal experience. And then I'm not discouraging any anytime any people can get chances, and that's
Yeah, that's great. I appreciate that you also emphasize that's your personal experience. Obviously,
all.
it's different for everyone and different times and different markets. I know there were a number of questions before the seminar just about international scholars, visas, things along those lines. So if you are wondering some of those questions, I highly recommend that you reach out to our Office of International Student and Scholar Services, or ISSS. I'm going to put the link to that in the chat. They are part of the university. They're here to help you with questions, especially if you're on an F1 or a J1 visa. They even have like appointments you can set up to talk to someone to get some more personal advice that's tailored to you and your own situation. So if you're wondering those types of questions, I definitely would recommend reaching out to that office because they are the experts there. Um, okay. Oh, we got two more questions in the chat. Okay. Our next one here is from Kate, and she is wondering, do you think having bench science experience is more of an asset in most industry positions, or is it position dependent?
No, definitely bench. Some people brag about bench to bed. They say like, "We I started my work this one, and then patient bed ended up going to the patient bed, and the drug or this product helping the patient on the bed." So definitely, our experience in the bench work is very important. And then that we'll from the skill set we can match some of the required skill set in the job description, and then we can find the job.
Yeah, that's great. I would add too, I'm not sure exactly what your research is, Kate, but I think some industry positions, obviously, if you're in less of a bench science field, like maybe you're in more of a computational field, obviously that would potentially vary drastically, um, the types of positions you're looking for.
Yes.
Okay, so we have another question here from Samuel. So he says, you said you made your initial move into industry via a contract job. For this, did you already know people working at the company to help you get a foot in the door? Also, did you specifically network to CDMOs, CROs, et cetera, to make connections to help get in the door? Or did you simply apply to an open application and get a job?
No,
Mostly, I'm trying to figure out the best ways to create networks to make transitioning easier.
yeah, it's through the network only. Actually, one of my known people, they were looking, their known people were looking for that contract job. Then luckily I came to know that. That's why I, and even though before I directly, I ended up talking with the hiring manager, and hiring manager also said like, this is a short-term job. You can use as a stepping stone. Then I thought, why not? We can try, give a try, and then, but still, that position was like analyst position. Then I asked for I built my resume differently, and then I showed like I have this many years of experience. I can start from the day one like that. Then I ended up getting a senior scientist, which is like five ranks different. So that's a huge leap I got, and then we through networking, basically through networking or through the people from the LinkedIn. Keep adding my people, whoever I know in the past, like 15 years back, 20 years back. I still, if I have something, I can go and reach out to them. Okay, he is there or something. So networking is the key.
That's great. Yeah, I like that you called out LinkedIn. We reconnected on
Yes.
LinkedIn. And for anyone who maybe if you're in the audience and you're not feeling super confident about your LinkedIn, we do have a LinkedIn workshop that's coming up in December. So if you feel like you need some support on getting your LinkedIn ready, it definitely is super helpful for networking and just maintaining those relationships. So definitely come to that workshop. It's going to be a virtual workshop as well if you are a remote student or postdoc.
Yeah, I would
Okay,
say
next.
we
Oh yeah,
can
sorry, go
make
ahead.
no, no. No, I would say we can always make use of the LinkedIn very good, even though lot of hiring managers or some of the people they are invisible in the LinkedIn, but still, where during the hiring time they will go and look into our profile, everything for sure. So always keep updating the LinkedIn, and then uh, if we are reshaping that whatever description we have all the time, if we are updating that, especially during the transition time, or if you are finding the next job, then we have to uh, update everything up to date, so that way that is the best place people look into it.
Great. Thank you.
Thank you.
All right, questions keep coming in. All right, we have another one from B, and she's asking, the lab I currently work in utilizes older pieces of equipment. How would someone stay on top of technological literacy if they want to transition from academia to industry?
No, even older piece of like when I started my PhD, I used to do enzyme-based cloning, so they are more classical way of thing. But we are learning from they were like even reagents. I had to make everything my own, so it's also will help us for formulation. It's even though old piece of experiment, I would say we'll gain something from there, and we can update our campus is not. We have very good resources like core lab. It's about even uh, some of the places I worked was I didn't have the resources, so I have to find out which lab has that resource, or I have to find out which one has more updated, or if core lab is there, how can I get support from my PI to reach that core lab facility, like small amount of money they have to pay. So we can do that, and even though old or new doesn't matter, uh, what we are getting out of it, it's more important. And I would say enjoy the research. That's all.
Yeah, that's great. Maybe this is my old old person bias, but sometimes I feel like if you've done it the older fashioned way, you might understand the technique a little bit more
Yeah,
deeply than if I mean, and I understand, of course, if there's new technology, you have to learn that too. But I feel like, part of training is being adaptable and learning how to learn new things. So
yeah,
if you're trained in the technique, I think that's got to be important.
yeah. I agree. The older way or classical way, like when we are doing the um, cloning, for example, old days, we have to pull up the sequence, we have to manually check everything. Okay, how we can mutate it, how we can delete this part. So if you know that one, then nowadays people are using Snapgene, everything. This is more easy, easy thing. So this is faster. We are getting that. done, but the older way is we know what we were doing. So everything has pros and cons.
Oh, Bruce. Bruce is raising his hand. All
I,
right.
I am making it official. I just want to make a comment to add to that. We had another alumni uh, presentation from Rick Sen, and Rick was talking about the technique that he used during his postdoc and how that was critical for stuff. And as soon as he transitioned to the industry career, nobody was using that anymore because it changes so quickly. And so I think the more important thing, as Hannah said and as Bala said, is really that you are aware of kind of what's out there, but you're able to comment on your adaptability, your ability to learn new things. That's going to be the more important thing because we're never going to keep pace with how quickly everything else is changing for it. And so being able to kind of tell that story at about times when you have learned new things or learned new techniques, that probably becomes the more important piece. Other thing, during our iSearch program, we had a career panel, or we did a site visit to this company uh, last semester, and one of the scientists there was talking about their first transition to industry from their postdoc and that they got hired specifically because there was this one technique that they used during their postdoc. And they got hired into that. And they said that once they started working in that position, they never once used that technique, that they got put onto a totally different project and everything was different. So of course, it's going to be position dependent. And there may be some positions that really need you to be doing that specific skill and all that. But really, being adaptable,
Yeah.
being flexible, being able to learn new things and come up, that's going to be equally important. So don't overemphasize, I mean, don't underemphasize, but don't overemphasize how important the specific technology is, knowing that you're able to learn new things. If nothing else, your PhD, your postdoc, your graduate training has demonstrated your capacity to learn. And so if you come in with that, you'll always be in a better position.
Yeah, I completely agree with you. That's like adaptability is most important. Like in the industry, like you said, only one. Like if they say for DDPCR, they want only for that one DDPCR, and then eventually, if they are moving to different technology, suddenly, like you said, it will be a different, a different one. We have to work on ELISA or something, or potency assay, something like that. So. That's why in most of the industry also they say embrace the changes. Like always we have to be adaptable to embrace new changes. Suddenly our priority get changed, so we have to stop that the current running one, and then we have to move to the next one because the rest of the other people they were waiting for that work to be done and then that product should be delivered for the patient or it should be ready for the clinical trial, something like that. So I completely agree with the Bruce comments.
I appreciate that you said that, Bruce, because I was thinking of that same story from Rick as well. All right, so we've got another question in the chat from Cassie, and she's wondering on the topic of networking again, to come back to that. Any advice on the best way to start building a network while finishing your PhD at CU Anschutz? So you talked a little bit about how you networked on campus. Maybe you could expand a little bit on ways that you networked sort of outside of LinkedIn. And if you're on LinkedIn, how should you be networking with people on LinkedIn?
LinkedIn, if we find any relevant job, like if it is, if someone is working there, if you know somebody like a second or third uh, connection, so we can still reach out to them and then we can add them. Sometime if we know the role and then if we are reaching out to them via uh, the LinkedIn first, and if we are applying after that, that will help a lot. I have some examples also, and like some of them they did that way, and sometime we don't. Many people they don't respond in the while working in the industry. Whoever working in the industry, they don't do too much time on the LinkedIn unless if they are looking for job, or if they are looking for person for a particular position, that time only they will go actively and look for that. So it depends. Sometimes it works. Uh, Approaching them, it's good to approach them first. If we don't get any response, no problem. We can submit the application directly, just to rearrange the resume based on the job description, and that will help us a lot. And for networking, any like I said, for example, if I don't have a resources in academia, I reached out to other people in the lab. That's also kind of a networking, and then we can also give us a networking even when they ask for, do you have any collaborative experience or networking experience or teamwork experience? We can give a lot of things from the academia, whatever we did, and I did that way, and then that also translate to the industrial settings somewhat good. So networking is important.
And I like that you kind of highlighted this at the end, but I think sometimes if you're a trainee, it's easy to overlook your own lab as a networking hub. So definitely ask your PI um, who they know. Also, like former people who have gone through your labs or alumni of your lab. Those are great people to reach out to because they have probably the most similar background to you. So don't forget about that as a potential place to look for opportunities. Um, just just a reminder. So,
Yeah.
okay, so that was all the questions in the chat. Um, Bruce, do you have anything else that you want to add? Or I can ask some questions that were from I feel like we covered actually most of the questions that were asked in advance, but I can look through this again really quick.
Yeah, and if you, I mean, I don't really have um, questions. I think Bala's done a great job of touching on, really those differences between academia and industry. I think, some of the things you said that really resonated with me were the emphasis on being part of a team and being adaptable, but also how we interact with our colleagues and in academia, challenging directly that Socratic method that we do when, somebody gives a presentation, it's like, oh yes, but in 1942 there was this study that you didn't, and that kind of stuff doesn't really make for the most cohesive team. And in an industry setting, that academic discourse needs to be a little bit different. And so I think that, those kinds of things about understanding the environment, I think that's one of the things that is so helpful when we hear from people like Bala who have made the transition and now are working in both spaces. So we can start to understand those differences that aren't really about the science. It's about the culture, it's the context, it's all of those kinds of things. So I think that really resonated with me a bit too. Not being so tied to that one project so that it's, where's the next direction going to take us and just kind of going down those rabbit holes, but being very focused on here's what we're here to do. This is the product we're trying to get there. We have to be very rigorous in this because it's not that academic science isn't rigorous, but it's different when that's something that a patient is going to consume. I mean, there are things that become different about it. So, I think it was great. I really appreciated hearing all of those and all of your insights on the question. So, Hannah, maybe I bought you enough time to have identified if there's one or two questions that we didn't hit. But otherwise, I think this has been really great.
Thank you.
Yeah, great. I don't see any other questions coming in the chat. So there was one question asked in advance that I think is a very nice ending summary question, which is just what would you say are your top 3 tips for getting that first job out of academia?
Yeah, first one is networking, and then the second one is communication. Like we have to communicate with the alumni or somebody who knows or somebody who is in they are in already who are in industry already, and then their friends, everything, and also we can also make use of the when we attend the conference. I personally after my presentation or post or whatever it is, I like to go to the vendor side and then I want to look what new technology is there, uh, what kind of new tools are there to bring to my lab or something like that. And then I try to talk with them, interact with them. I try to get connection. So if we are breaking the ice there and if we are getting a good rapport with that people who are, even though it is a vendor show, but they will be definitely helpful. They are also great scientists and we can reach out to them, we can keep in touch with them, and sometime who knows we can find a job in their place also. We can approach them. So uh, I use make use of networking, communication, and like communication, like not criticizing directly, just a polished way of communication. Like first you appreciate the people, then criticize the people. That's what I learned. Otherwise, in the PhD time, we learn like we have to criticize each and everything, every data, everything. But industrial setting or approaching a person and from the industry, it's like first making a connection, uh, then we can he will definitely help you if we make the connection with the people, he or she definitely. So networking, communication, and then like make use of the available resources.
Yeah, that's great. I appreciate that plug to resources. Um, yeah. Okay. Well, if there's no other questions, we can go ahead and wrap up. So, Bala, thank you so much for your time today. I really appreciate it. I know the audience appreciates it as well. If you are here, I highly recommend you practice your networking skills by reaching out to Bala on LinkedIn and making a connection and saying thank you. And we hope to see you in the future at other CDO seminars. All right,
Yeah,
thanks
thank
all.
you. Thank you, Bruce and Hannah. It was wonderful meeting you, talking with you, and thanks for everything. I will keep in
Yeah,
touch.
thank you.
If any of you have any question or anything, you can reach out to me, email or via LinkedIn. Good to see.